Good morning, partner. Got an identification on this one?
It's Rose amphibious.
Get the cell out for me, will you?
Sure thing, detective.
The blood pool is indicative of a high drip rate and messy weapon choice. Perhaps a serrated knife or point-blank gunshot? The wallet seems to be missing as well. Behind door number 2 is another dead body. No blood pool, but blood dripping from the mouth suggests internal bleeding. Looks like she could have choked on her own blood. The wallet was also stolen. Oh my god. We have to stop meeting like this, partner.
The wallet is missing again, but there's barely any blood.
Elementary, my dear partner. See a very small incision on the neck. Probably used a very sharp object. Blood drip rate is very slow, so we know he didn't hit the artery. But nevertheless, she's still dead.
Hello, we're Group 11. We hope you enjoyed our trailer. The disembodied voices you will hear will now explain our experiment and the detective's findings. The 3 drugs pictured here, high potassium, dopamine, and angiotensin II, were perfused through a frog.
Here is Starling's equation of ultrafiltration. This states that the flux of fluid across capillary walls, phi, is equal to forces out, P sub C plus pi sub I, minus forces in, P sub I plus pi sub C, all multiplied by the filtration coefficient K. Forces out is made up of capillary hydrostatic pressure, P sub C, and interstitial oncotic pressure, pi sub I. Forces in is made up of interstitial hydrostatic pressure, P sub I, and capillary oncotic pressure, pi sub C. Furthermore, a high K means that it is easy to cross a capillary wall, and a low K means that it is difficult. This equation can be used to explain weight changes in the frog. Here is Ohm's law. This states that flow, Q, is equal to the change in pressure along the tube, delta P, divided by resistance to flow, R. Resistance can be described by this equation here, with eta as the viscosity of the fluid, L as the length of the tube, and R as the radius of the tube. These equations can be used to explain drip rates in the frog.
The name of the first victim is Rose amphibious. She was killed by hyperkalemias, which is high potassium. Her wallet was stolen, which indicated a weight loss from 0.030 grams per minute in the control to 0.008 grams per minute with the high potassium treatment. There was a large amount of blood pooling, indicating an increased fluid drip rate of 28 drips per minute to 30 drips per minute.
Before we go over the effects of increasing the extracellular concentrations of potassium, let's first discuss what normally occurs in smooth muscle cells. Under normal conditions, there are quite a few channels through which potassium can move through. The 2 we will focus on are the sodium-potassium ATPase pumps and the inward rectifying potassium channels, labeled here as KIR. Sodium-potassium ATPase pumps transport 3 sodiums out of the cell and 2 potassiums into the cell with the help of ATP. This results in a net efflux of cations. Inward rectifying potassium channels transport potassium out of the cell and into the bloodstream along potassium's concentration gradient. The main ion responsible for any kind of smooth muscle contraction is calcium. When L-type calcium channels are activated in response to depolarization, calcium ions are allowed to migrate into the smooth muscle cells. Once calcium has entered the cell, it is involved in the sliding filament theory. First, calcium forms a complex with calmodulin, labeled here as CAM, in a 4:1 ratio. This complex then activates MLCK, myosin light chain kinase, that is responsible for phosphorylating the myofilament myosin. This myosin is then able to bind to actin and form cross bridges and power strokes involved in vasoconstriction. MLCP, myosin light chain phosphatase, is the counterpart to myosin light chain kinase that dephosphorylates myosin, thereby inactivating it and not allowing for any contractile activity. However, increasing the extracellular potassium enhances both the activity of sodium-potassium ATPase pumps and the inward rectifying potassium channels. The activity of sodium-potassium ATPase pumps is increased because there is a greater driving force for potassium to enter the cell. The inward rectifying potassium channels are directly activated by potassium that results in a potassium-induced potassium efflux through these channels. The combined effect of the enhanced activity of these two channels is a net efflux of cations. This results in a hyperpolarizing current that inactivates L-type calcium channels and therefore causes the opposite of what we discussed, which is vasodilation. Now let's see how this relates to our results. Vasodilation at the arterioles means an increase in the radius of the blood vessels and therefore a decrease in the resistance. Decreasing the resistance equates to an increase in the flow and therefore an increase in the drip rate. Though there is vasodilation at the arterioles, the vasodilation on the venous side is much, much larger. This net vasodilation of the venous smooth muscle cells decreases the capillary hydrostatic pressures by reducing the blood volume present at the capillary site. Since the net movement of fluid from the interstitial tissue back into the capillaries is now greater, the frog begins to lose weight.
The name of the second victim is Julie Hopskin. She was killed by Larry L. Dopa, which is dopamine. Her wallet was stolen, which indicated a weight loss from 0.014 grams per minute in the control to negative 0.023 grams per minute with the treatment. There was no blood pooling, just a small amount of blood dripping from the mouth, indicating a decreased fluid drip rate of 28 drips per minute to 26 drips per minute. The third victim is Anne Fibian. She was killed by Angela Tensin II, which is angiotensin II. Her wallet was stolen, which indicated a weight loss from negative 0.007 grams per minute in the control to negative 0.011 grams per minute with angiotensin II treatment. There was barely any blood, just a small drop on the neck, indicating a decreased fluid drip rate of 30 drips per minute to 22 drips per minute.
Dopamine is a neurotransmitter belonging to the group called catecholamines. Dopamine can bind to different membrane receptors that can activate various G-protein-coupled signal transduction pathways. The 5 dopamine receptors are D1R through D5R. Dopamine can also bind to alpha-1 receptors as well as beta-2 receptors.
Angiotensin II is a peptide hormone that can bind to alpha-1 receptors. It has 2 main G-protein-coupled receptors, AT1 and AT2, and these receptors can initiate the GQ signal transduction pathway.
When dopamine and angiotensin II bind to an alpha-1 receptor, the GQ protein associated with the receptor is activated and then goes on to activate phospholipase C, or PLC, which is an enzyme associated with the membrane. PLC activation leads to the conversion of the membrane PIP2 into diacylglycerol, or DAG, which resides within the membrane, and IP3, which moves freely in the cytosol. DAG activates protein kinase C, or PKC, in the cytosol, which then activates L-type calcium channels, or LTCCs, and inactivates potassium channels. Thus, PKC causes calcium to flood into the cell and prevents potassium from leaving the cell, prolonging depolarization so contraction can occur. Increased IP3 levels also exerts its own effects by activating IP3 receptors on the sarcoplasmic reticulum, a specialized endoplasmic reticulum within the smooth muscle cell that stores and releases calcium. Activated IP3 receptors release stores of calcium into the cytosol. The abundance of calcium ions in the sarcoplasm creates the calcium calmodulin complex, as we explained earlier. Because dopamine and angiotensin II cause a decrease in drip rate, we concluded that the arteriole is constricted. Vasoconstriction causes resistance to increase, leading to a decrease in both Phi and drip rate. Resistance increases because the radius decreases during vasoconstriction. Since dopamine and angiotensin II vasoconstrict and increase resistance, they reduce the flow of fluid into the capillaries by decreasing P sub C, lowering overall forces out, and therefore Phi.